What's up, everyone? It's your boy. We back, and we're back on something special. It's been a while since I done this. Bioinformatics. I need to understand the motive scan. What did I do with the motive scan? Let me see. Let's see. Let's bring in GPT and Gemini. All the AI is coming in. Wow. She. Oh, I did not got rid of the. Excuse me. Excusez-moi. Excuse me, moi. Refresh. There we go. The chat is gone. It's not like that though. Emir Sekola. Yo. Yo, what's up? What's up, dog? What's good? What's good? So if I remember, you're back. What? Did you come in today? I mean, I did not notice your name today, but it's not your first time checking out the stream, okay? I'm sorry, gang. I don't remember the conversations we had before. What you going to play? Ah, man. I think I'm gonna disappoint you, gang. Because I'm not gonna play any video games. I was working on something. That's fine, be you. Oh. Whew. Appreciate that. Channel activity. Oh, yeah, you're the one that got hit. Okay. No. But hey, at least you recovered. And, it's a, let's just say it's an experience. What year are you in? Second year. Second year of my university. Zweite Jahre. Ja, genau. Okay. $100. Uh, nice. What age are you? I am 22. Zwei? Yo, if you had one wish, what would it be? Anything you want. Damn. What? What's damn about it? Okay. If I had one wish, it could be anything. Oh, you're 10? Yo, so you got hit when you were like around that age? Mm-hmm. That's good. It's good fun. How does it then you anything I want. You were 6, damn. You're 3-year-olds right now. Mm-hmm. Oh fuck, what could I wish for? Like whatever I'm thinking is gonna lead to I don't know this hypothetical singularity. Like if everything's perfect, then there's no such thing as living. Then you subbed? Respect. Nah, me. I don't know. I don't know if I could wish for anything. It's so weird. I don't know how to answer that question. I mean, I could say something like, "Oh, I would like superpowers or some shit," but come on. What's my religious background? Oh, that's a good question. I like it. Like people ask, "Oh, what religion are you in?" So basically, I was brought up as Christian. I went to church type shit, but now I'm not. I mean, when I was with my parents, I used to go to church and stuff, but now. What month were you born? Oh, it's March. worked on GCNR. Can you let me know what's workflow I have? I have to work on the rest of them. Why don't you study all religions? Because I don't know if I have time for that. Maybe in the future, if I feel like, oh, I want to learn how Hinduism is working, I want to learn how Muslim religion is working, or I would want to learn how Buddhism is working. I want to learn how fucking, I don't know, other religions. Maybe you get the answer for everything. I don't think so. Maybe I would. Maybe I would. Who knows? Raw bamboo camera analysis, count every possible 6 merge, compare enrichment versus random expectations, examine known biological motives, global frequencies only provide a rough picture, position is required. Motive scan. Face, poop, shape, face, red, smiling, live, volcano. I don't know. What's that? What you have already finished, understanding the bamboo genome. What date in March? 16th. Yeah, try sometime when you get time or after your exam. Yeah, maybe I would. Nice. In that case, I should also try reading the Constitution. That's also something like a really big book, right? Sure, yeah. Pie charm. Oh, god damn, it's been a while. I don't even remember what I did. God damn it. Were you born in 2004? Yeah? I just calculated that in my head. That's good. That's cool. Atheistic ideology right now. I wouldn't say atheistic, but boring. Boring in what terms? About the atheistic ideology. I do not believe that, oh, there's no such thing as a God, but there is something that makes all of this possible, right? And you could name that unknown thing as God, maybe. I mean, there's always things that we don't know. And if you If you collect everything that you don't know, and you could name it God. What is this subject? It's bioinformatics. Basically, we're using computer to analyze, understand biological data. You're smart. We're all smart. We're all dumb and we're all smart. This is the motive scan program that I tried running. I don't remember why I ran this or what the result was. I do remember the answers, the result from the Kamer analysis was not that useful. So that's the reason. That was I moved to motive. I actually think I know exactly what you wrote the script. Looking for the GCN. This is just next script. It was your attempt. It's also shut the fuck up. I don't care what I was doing. Just let me know why it did not work out. Your conclusion was Kramer analysis tells me what sequence exists and how often, but not where they occur. That doesn't answer. Are they clustered together? Are they near a gene? The answer was no. So you can. Mm-hmm. One camera setup is less, the one showing your head. So I use the desk. That's where I put the camera here, and you see my desk. That is usually involved when I'm writing something, when I have to write something on my tablet or something, or on paper. Right now I'm just typing. I don't think it's useful. I'm working on screen right now, so. Matthew Legend, hello brother. You back from lunch. Don't you have physics as a subject in your course? Yes, I do. I did pass physics. That's fortunately I did pass. What's the script you're writing? Right now I'm working on. I actually still not sure what I'm working on, but I'm trying to figure out what these different methods we use to analyze genetic data. I still don't know how these work. How are we analyzing it? So I'm trying to figure out what are these things that we do. So it's more like a self-study more than working. Okay. Aren't they clustered together? Are they okay? Right. Again, I need to see why was Kramer analysis not working. What did I write? Slide a window of length 6 across the entire genome. Count every possible 6-letter DNA. Enrichment tells if certain sequence is biologically favored, as 4096. Okay. Count all the 6 mers. Calculate enrichment versus random expectations. Checks specific biologically known motives. Yeah, I don't understand these things either, but I hope you're gonna learn it eventually. Yeah, that's how we learn, right? We try different ways, and after some point, we're gonna start seeing some patterns. Camera analysis revealed that most biologically known motifs show the weak enrichment compared to the random expectations, while repetitive sequences dominate the genome. This suggests that the global motif frequency alone is insufficient for your gene identification, and positional motif scanning is required for functional interference. Positional motive scanning. Okay, that's where motive scan is coming. To improve spatial resolution, the gene can be divided into fixed size windows and camera frequencies recalculated per region. This allows detection of local enrichment patterns that are hidden in global averages, although it still does not provide direct gene level annotation. Camera analysis is alone not sufficient for biological interference because it only provides a rough global statistical picture of sequence composition without positional or functional context. So it was not that useful. Camera analysis was not that useful. Now let me know why I should use motive. Instead of counting motifs globally, I'll locate every occurrence in the genome. So the question changed: how many Ts exist to where all the TTA sequences? That's more biologically meaningful questions. Look through your code. Let's go through logically rather than line by line. Motives. This is your search list. These motives already have biological meaning. You aren't searching every possible sexmer anymore. You're searching motives you care about. And my brain is not coming up with what exactly is a motive. It's an ad, fucking ad. Was an ad. Oh, I changed it. Okay. Didn't know that. But if I share my screen, I feel like it's gonna even use more power. I hate that. Loads one chromosome per scaffold at a time. So instead of treating the genome as a one giant DNA string, you're processing each chromosome individually. Slides across a chromosome, one nucleotide at a time. Exactly like the camera analysis, okay? Except now you're counting every sequence. You're asking one question repeatedly. Does the DNA here equals result motive? This is important improvement. Instead of now you obtain then you can immediately notice Let's solve some physics. Then if you have the paper simultaneously, you and all of us here, there's only me, lol. Yeah, we could solve physics, but right now I was looking into this one, bro. I mean, I haven't looked into this self-study for a long time. I need to get back on this. I was busy with other subjects. Not like others are not important, but this is also important to me. If you want, I could give you the physics papers. I don't know how I will send it to you, but yeah. Now you found two biologically related signals that are very close together. You couldn't do that with camera analysis because it has discarded all positional information. Okay, so motive scanner is mainly mainly saves the position of the specific sequence. Biggest conceptual jump. Oh, junk. I think this is the sentence that connects your workflow. Statistics. You're literally building a map of genome, not a gene map, a motif map. Why these motifs? Notice that your list isn't random. Okay. These are all motifs related to plant development and regulation. So you're asking where in the bamboo genome are regulatory. Okay. What was the output? From what I remember, your script generated a file called motifscanresults.txt that looked like, yeah, the file later became the input of your ORF finder. So why did you move on? Because this was not enough. Okay. Let me find the motive scanner report. Yeah, there you go. Motive scanner result. It's a big boy. That's a big boy. Look at this. God damn it. Woo! So total hits. I'm guessing these are positions. First chromosome. Okay, nice. Yeah. Can this is what the first few lines from the motive scan program gave me as the result. Let me know what this exactly means. I'm guessing the 3-digit numbers or higher are the positions. And could you let me know what the one is? I thought it was the chromosome, but looks like the whole file is just having yeah. So I'm guessing, Matthew Legit, you are using the vertical stream. The vertical stream is only showing half of my screen, half of my desktop screen. The other half is on the left side, or yeah, this would be left side, right? Yeah, on the left side, and that's not visible. and ChatGPT is on the left side. That's where I'm typing and stuff. I thought it was a Pro, but it looks like the old file just... Also, if I understand motive... Yeah, I'm using the like the YouTube shorts one. Yep. Yep. And if you want, I could change it. I could change it. Sequence and marks its position. Also, could you give me a brief explanation on the name itself, because I need to... What the CMO, this is not chromosome 1, this is the ascension ID, assigned to one sequence in the reference genome. Remember this line in your code, chromosome equals record.id, comes directly from the FASTA file. If you open the FASTA file, you see headers similar to... So your program simply stores whatever identifier NCBI gave that sequence. Again, are we talking chromosome or identifier inside the chromosome, inside each chromosome? Is it a chromosome? Usually yes, for high quality assemblies. Each CM ascension corresponds to one assembled chromosome. If you think of important thing is that they are unique DNA sequence in the genome. Do that number change? It's still 4, 5. Oh, okay, there we go. We got 4, 6. Okay. I see others. 4, 8. Okay. Back to 4, 9. What's the last part though? Okay, I don't know what the last part is. There's no need to store both a start and end position. Your code only stores the starting position because the motive length is already known. Those 6 spaces are okay. Why the starting position? Suppose your motive is length 6. If the scanner reports 1000, you immediately know 1000. Okay. The ending position is simply, because the motive length is fixed. What exactly is a motive? This is important biological definition. A short DNA sequence that is known or suspected to have a biological function. So what is Kamer? What exactly was Kamer? Slide a window of length, no. Count all 6 mers, calculate enrichment versus... So Kamer and that... Kamer is also 6... if this is the definition of motif, let me know what exactly is Kamer. Somebody use the chat. Give us a room tour, brother. It's like the smallest room out there. And it's pretty fucked up. I could give you a room tour just a little bit later. This shit looks ridiculous right now. Good question. The difference between Kamer and motif is actually the key reason why you moved from Kamer analysis to motif scanning. They look similar because both are just... Did I just waste my time reading the first part? Any DNA sequence of length K extracted from a genome. Okay, Kamer is a more broad term. Okay. It's a more broad term. Where do I sleep? Right there. My bed is right here. My workplace is right here. My window is right there. The washroom and everything's up front. The kitchen is like common kitchen. It's outside. It's not in the room. It's outside. You Christian, right? I don't go to churches. But on paper, like if you look into the government documents, yes, it says that I am of religion Christianity. But I don't go to churches, I don't read the Bible, like nothing. I need to add this into my freeform. How do I add it? How do I add it? Motif scan. What was it? Motif scanning searches genome for predefined biological meaningful DNA sequence patterns and records their That's a motif scanner, okay. That's almost exactly right. The refinement I would make is motif scanner scanner finds predefined biological meaningful sequence patterns and records the genomic coordinates where they occur. Okay. Damn, I'm starting to get hungry. God damn it. Fuck. I hate. Sometimes I hate being hungry. Damn, I do wish we had some sort of tablet or something that you can just swallow and it gives you enough calories, proteins, every required thing to survive a day with caffeine. Come on, man, we should be able to figure that out. It's not that hard. Okay, now I understood that part. Let me know, how do I proceed? Like, I got the results, and what do I understand from the result, or are we moving to the OR finder? Neon DOS. Hey, bro. Oh, I'm like 5 FPS. What was that? What's up? What's up? Neon DOS. Not gonna lie, I don't. Oh, you're not on the vertical stream. You're on the. No, you were on the. My computer is getting slower. Like, get the hold of yourself. Come on. Like, I'm not even running anything that that is energetically expensive. I'm not running that much. You're in college, right? Still procrastinating. Can I join your stream? Oh, you're the one who asked me about suicide. Oh, okay. You were the one. I accidentally mistook someone else. Oh, you were the one. How you doing, bro? I hope everything's going good. Or finder. What you understand from the motive scan result, your current result, TATTA box, gene start signal, total hits. Your first interpretation is bamboo genome contains many locations where the this DNA pattern occurs. This is all you can conclude from this output alone. You cannot yet say this is a gene, this is a promoter, this is a control growth, because TTA is short and can appear randomly. The problem now you encounter is your result is actually showing the limitation of motive scanning alone. If you investigate every single hit, you have hundreds and thousands of candidates, too many. So the next question becomes, how do we reduce these millions of motive hits into biologically meaningful regions? This is where Nox plus TATA plus ORF logic comes in. Next step is ORF finder with combined with motifs. You don't simply run ORF finder with the single whole genome. The genome is too large. Instead, you use motif locations as starting points. Hey, yo, what's up? Genome is too large. You use motif location as starting points. Check nearby regions upstream. What an ORF finder is basically a DNA sequence that has a structure needed to potentially encode a protein. It looks for start codon, stop codon, example, could potentially become, okay, so why ORF comes along after motif scanning, because the combination is much stronger. Compare only motifs, probability of being meaningful low. What are you studying? Right now I'm studying basic tools used in bioinformatics. I'm trying to figure out what are these tools used in bioinformatics doing? How is it working? It's a sort of self-study, you could say. So motif plus ORF gives probability higher. What you should add on to your work: motif scan identifies genomic locations of regulatory motifs. However, the motif occurrence alone cannot confirm gene regulations because gene regions, because short sequences can occur randomly. Therefore, the candidate regions are further examined for open reading frames representing possible protein coding sequences. Again, I still didn't fully understand ORF, open reading frames. How is it gonna save time compared to the motif result? I need to add this on my workflow. What? You live in Austria, right? Yes, correct. Oh, I forgot. You could see my location on YouTube channel, right? Yeah, that's a good way to find out where is this account based on. Yeah, you should go to more at the very bottom. Yeah, they show it's Austria. You told me before when I asked. Oh, okay. Yeah, I remember saying that. But you could also see it in a previous live stream. Okay. But there, yeah, you could also know it from my channel. That's also a good thing. Yo, by the way, now I'm only seeing the ChatGPT. So okay. Yeah, I mean the code part. I'm not doing anything. I'm still here. Oh, Matthew Legit, you're still here. Well, I found the person who I accidentally mistook as you. When you came up to my stream, I asked you like, You are the one who asked me about suicide, right? I remember a conversation happening between us, but it was not you, it was this guy. Where is it? Was uh Neon Dust. That was the person who asked about suicide in the chat. Okay. I understood why proceeding with the results from the motive is not very productive and very time consuming, but I still didn't understand how the my spelling is wrong, how the ORF is gonna save time. I mean, I was talking about it with you too. Yeah, that's after I told you, right? But yeah, don't worry. Don't worry about it. Don't worry about it. It's actually the most important question because it explains why pipeline. Find every TTA location of the problems. A 6 base pair sequence is extremely short. A random genome will contain many short sequences. This location contains these patterns. It does not. Okay, yeah, ORF asks a completely different question. Does this region contain a sequence that could actually produce a protein? A protein coding gene has structure. Oh, it has a start and end. Okay. An ORF finder searches for. Okay, why this reduces candidates? Imagine your motive scan finds location. Now you check each location. Yeah, you said Bollock, the code word. Emir Sokola, I was looking for your stream. Now I'm back. Yo, what's up? Is it hard to find my stream? YouTube, what are you doing? I'm not thinking about it for a long time now. Don't. Or ORF is basic. Okay. So let's start with the motive where it finds specific sequence of finds specific sequence and ORF checks for start and end codon between the specific sequence that motive found. How is the sequence 6-mer sequence that the motive located gonna be involved in the ORF finder? Hey, watch the music video of a song named Tabahi. Bro, please don't tell me it's some OnlyFans bullshit. This is exactly the point where the workflow can become confusing because the motive and ORF are not same thing. They're two different types of information that are combined later. My YouTube is slow as fa. As fa. Tabay. Oh, it's from the movie Toxic. Is it a good music or what? No, it's from Retro Panda. What? Which are you mentioning, Vibeof? Is it from the movie Toxic or what? Toxic. I heard this movie. What the motive scanner finds you? Your motive scanner finds regulatory signals. Genome. ACGTAC. Nox motive. TATA start stop. Wet. Looking for motive. It receives a region of DNA near the motives. It receives a region near the motive and asks, this is positive. Okay. In that case too, it has to go through all the motives, right? Even in that case, the program has to go through all the sequences that motive separated. But the important difference is how much sequence it has to search. The ORF finder does not go through the entire bamboo genome. What motive scanning first? Your motive scanner already gives you a position, okay. Now ORF only checks around these locations. Visit each location, okay. So that's what ORF finder does. So let me know if this uses the FASTA file and the motive result to check valid sequences. Protein sequencing. Oh, what the fuck is this? You see that? No? Nobody saw that? Hello, bro. Yo. Yes, your understanding is mostly correct. This ORF finder uses both. That combines them to search for possible protein coding sequences, but one important correction, it does not directly check if the protein sequence is valid. It checks if the DNA sequence has a structure of the possible protein coding gene. The BLAST step later checks whether that predicted protein resembles known proteins. That's where BLAST comes in. I was not able to run BLAST. I remember the place. Okay, best friendo. I hope you're a better person now compared to before. Matteo. Yo, why do you care about others though? Okay. We're about to reach 160 subscribers. What? 160? Yeah, 160. I'm hungry. Okay, I'll stop with the ORF finder. I'll run blast next time. FNA path, where is it? Oh, whoa, this is a pretty big one though. FNA path, motive result path, upstream window 200. Just a minute. Hey, do you have to hack? Do you have to hack? What? What do you mean, do you have to hack? I don't have to hack. Oh, look at this. It's full of mistakes. 4 errors, 13 warnings, 16 weak warnings, 64 typos. That's what you get if you let AI code. Okay, bro. Okay, bro. Motive scan results. The file contains locations like Nox or your ORF finder reads this file here. The function extracts, meaning on this chromosome, Nox-like motifs occurs at these positions. Go to every Nox location. Now the program goes back to the original DNA. Check upstream for TATA box, your code. And the program goes to the original DNA. Your code, you're setting upstream window to 100. It means look 200 bases before the Nox motif. Okay. Your code asks if the motifs in the upstream, does this region contain any of those? If not, discard because it does not have any expected promoter signal. Search downstream for ORF if the TATF is found. Downstream sequence, meaning this takes the 500 bases after the Nox motif and looks for stop codon. What time do you stream until? I'm gonna stop the stream like within 10 minutes because I'm pretty hungry. I gotta eat something. I can't keep on drinking water. Maybe a little bit of this. Okay. So 900 base pairs keep, but 60 base pairs discard. What does the output represent? If everything passes, you save candidate append into. This is the candidate gene sequences. So you're full. Oh, okay, bro. I'm still here. Yo, respect. What do you like? I'm gonna be until the last second. Oh, what do you do? Like, you're also preparing for some exams or something? Paris. Do you play any games? No. Okay. So you're working on something else, but you have the stream on background. Respecto, respecto. Yeah, I mean that's what you have to do. You could also work on your stuff, right? Kivu, like you asked, do I play any video games? I do play any video games. Right now, I have two. I have option to play one of these two games: Sleeping Dogs or Lies of P. Let me know what can I play. What should I play? Are you working? Kind of, yeah. I'm studying, which you could consider as work. Motive scan results. There we go. Okay, bro. Like I asked you, should I play Sleeping Dogs or should I play Lies of P? Bye, I'm going to sleep. Okay, you just ignored my question. Well, respect for checking out the stream. I don't know, bro. You choose. Okay. Yeah, I was asking he Hivu. I was asking Hivu, like, bro.